Plasma membrane. It is also called as cell membrane or plasma lemma also. It is selectively permeable in nature. It can be studied in two different versions, as early study and later study. In the early study, we will talk about lipid bilayer model. In the later study, fluid mosaic model we can take. Let's talk about the first model. The first model, or as the early study, that was given by James Danieli. And this study, it is called as a chemical study. It's a chemical study. And this was given on the study of RBC cells. And this represents the model of lipid bilayer. Lipid bilayer model. And according to this lipid bilayer model, the lipids are arranged in a bilayer or else in two layers. Here you can see over the top and over the bottom. And here this bilayer of lipid that consists of head and it consists of tail. Head is towards the peripheral side and tail is towards the center. So here head is present and this head is towards outside. It is hydrophilic in nature. Hydrophilic, water loving in nature, or else it can be considered as polar in nature. And coming to the next, to the center, it consists of tail, and this tail is towards the inside and it is hydrophobic in nature, or else it is considered as non-polar tail region. Now, due to presence of this lipid bilayer condition and the tail filaments, that shows a flip-flop kind of movement. It shows a flip-flop movement. Flip-flop movement is a property of lipids or proteins. If a question is asked like this, the answer is lipids. It's a property of lipids. Likewise, we'll go for the second model. Now, if you talk about the second model, now this model is called as a biochemical study. Biochemical study. And this biochemical study, it is also undergone on RBC cells. And this was given by Singer and Nicholson. And it is considered as the most widely accepted model of plasma membrane. And this model is called as fluid mosaic model. Fluid mosaic model. And whatever this fluid mosaic model is there, it represents more composition about plasma membrane. And here particularly that was well studied on RBC cells, we said. So if you look at the composition, it has two different kind of categorizations. Major composition. If you look at the major composition, that includes proteins and it consists of lipids. And the major or else the common type of lipids which are present in the plasma membrane, they are phospholipids. And coming to the minor composition. Now if you look at the minor composition, the minor compositions that includes carbohydrates and it also consists of cholesterol. So these are the two different compositions or categories of compositions which are making the plasma membrane property. So here is a question. What is the distribution of proteins and lipids in the mammalian RBC cells? So here, if you talk about the human RBC, in case of human RBC cells, proteins that makes up the major composition, which is about 52%, and here lipids that makes about 40%, and rest, that's a minor property that is present, which includes carbohydrates and cholesterol. And here lipids are the one which are responsible for that semi-fluid or quasi-fluid nature of plasma membrane. So here the quasi-fluid nature of plasma membrane is due to lipids. If a question is given, flip-flop movement. If a question is given, quasi-fluid, the property belongs to the lipids. Okay, so let's look at the type of proteins which are present. So the proteins, they are of two types. They are intrinsic and number two, that is extrinsic. Intrinsic proteins, they are embedded inside the membrane. Extrinsic proteins, they are present on the surface. That is why extrinsic proteins, they are also called as surface proteins, and these are easy to remove. Coming to intrinsic protein. Now these proteins, they are very important. They are present embedded inside, due to which they act as transmembrane protein, or else they are acting as tunnel protein, where they help in transport of substances. You can observe that one here. There's an intrinsic protein, and over here there's a surface protein. And this intrinsic protein is the one which is responsible for the transport of substances from inside to outside and from outside to inside. Now, because of this lipids nature, the proteins are able to move transversely inside the plasma membrane. So here, what's the point? Transverse movement. The transverse movement of proteins, they are due to fluid nature of lipids, right? The transverse movement of proteins are due to dash nature of lipids. So what is that nature of lipids? The fluid nature of lipids. Let's talk about functions. Now if you look at the functions, as the plasma membrane is semi-fluid in nature, or else as it is, right, flexible or else elastic in nature, it can help in growth of the cell. So it helps in cell growth. From both the sides, now this plasma membrane can invaginate. As it invaginates, this invagination grows and reaches to the center and helps in dividing the cell into two pieces. And this property, we call it as cytokinesis. It means it helps in cytokinesis, which leads to cell division. Cell division. Now, it also throws the materials out, and this property is called a cell vomiting process, or as exudation, or else excretion process, or else we can call it as exocytosis. X means outer, right? So this process called as cell vomiting process. Not only throwing the materials out, it also takes the material inside, and this process called as uptake, or else cell eating process if it is a solid material. If it is a liquid material, it is called as cell drinking process, and this is also called as endocytosis. Now this endocytosis is further again of two types. If it is a solid, it is called as phagocytosis. So what is phagocytosis? The intake of solid materials by plasma membrane, the process called as phagocytosis. If it is a liquid material, right, then that is called as pinocytosis. Pinocytosis. And this only we call it as cell drinking process. And here phagocytosis, it is called as cell eating process. Cell eating process. Now, apart from these functions, it also helps in transport of substances. Now, if you talk about the transport, number 5, that is transport. Now, this transport can be of simple diffusion, or else it can be of active transport also. So it is of further again two types. It can be passive transport, or else it can be active transport. If it is a passive transport, now this passive transport, right, it is a transfer of substances from high concentration to low concentration along the concentration gradient. So transfer of substances along concentration gradient, and it is from high concentration to low concentration. So here no ATP is required, and here again two types are present. Number 1, if it is without involvement of protein carriers, then that is called as simple diffusion. So what is simple diffusion? Transfer of substances along the concentration gradient from high concentration to low concentration without using ATP and no proteins that are involved. So here there are no protein carriers involved. No protein carriers that are involved. Coming on to the second. The second one, it's a facilitated diffusion. Facilitated diffusion. Now, if you talk about this facilitated diffusion, it's a transfer of substances along the concentration gradient from high concentration to low concentration without using ATP, but in presence of protein carriers, by the use of protein carriers. So protein carrier are involved. So this is about passive transport. Coming to active transport. Now this is quite opposite when compared to passive transport. Now this takes place against concentration gradient, and this takes place from low concentration to high concentration, and here ATP is required. Now whatever this process there, the best example for this one, that is Na plus and K plus pump in animal cells. Na plus and K plus pump. So this is all about the plasma membrane. In the next session, we'll go with endomembrane system. Thank you.